Zachary is an associate professor and head of the neuroimprinting laboratory of the Institute of Biotechnology and Biomedicine at the University of Valencia. Her research is focused on the identification of molecular mechanisms that regulate neuronal stem cell biology and epigenetics of genomic imprinting in the adult brain. Her research group has described for the first time relaxation or absence of genomic imprinting in somatic stem cells as a new mechanism of regulation. Her studies have also defined the role of methylation and hydroxymethylation in brain function and during brain tumor formation. Her research group belongs to the Spanish National Network of Cell Therapy, Retic Tersal, and also CIBERNED. And the title of talk today is Genomic Imprinting in Neuronal Stem Cells, the Regulation in Human Syndromes and Brain Cancer. Zachary, thank you, and you can share your screen when you are ready.
Okay. Okay, can you see it? It's okay there, sharing?
Yeah.
Okay.
Yes, perfect.
So first of all, I want to thank the organizer of the CIBERNED for this invitation. I'm very happy to show for the first time in the CIBERNED the work that we are doing in my lab. We belong to the unit of neurobiology at the University of Valencia, also with Isabel Fariña, and we are one of the groups in the CIBERNED. So today I'm going to talk about genomic imprinting, and I'm going to focus on more specifically in brain cancer, and I'm going to show some of the work that we have done recently on defining how this molecular epigenetic mechanism controls brain cancer. I'm going to put just a moment. Okay. So genomic imprinting I'm going to make an introduction to talk a little bit about the genomic imprinting process, and it's an epigenetic mechanism that causes genes to be expressed in a parent of origin manner. That means that if normal genes express only by the two parental chromosomes, these normal genes express the two copies of the two parental chromosomes, and they show biallelic expression. Imprinted genes are only expressed by one of the parental chromosomes. This monoallelic expression is important for the normal development of the fetus and also for brain function. When in pathological condition, when two maternal chromosomes are inherited, what happens is that, for example, if we talk about gene A or gene B, in the maternal uniparental disomy, what we find is that gene A, we find no gene dose of no expression of gene A, and we can find a double dose of gene B. Or on the other side, when paternal uniparental disomy, so the inheritance of two paternal chromosomes, what we can find is that no gene B is expressed, and we find a double dose of gene A. For normal genes, this wouldn't be a problem, but for imprinted genes, it's very important that a monoallelic expression is maintained for a correct development of the embryo. This imprinting control is made by the ICR, or imprinting control region, which is a differentially methylated region, or DMR, that is regulated by DNA methylation, microRNAs, and histone modifications. All these marks are known as imprints, and these are established in the germline. After fertilization, the two parental chromosomes maintain these imprints, this methylation of these microRNAs or histone modification in their genomes. And this imprinting states is maintained during the whole development. And this is happening in somatic cells until the adulthoods, but is erased, these imprints are erased in primordial germ cells to remove imprints and re-establish new imprints during gamete formation. In the male, paternal imprints are established just before birth, and in females, these maternal imprints are established after birth, and all of them are maintained until adulthood for a normal development of the organisms. So far, only 200 imprinted genes have been described in mammals. However, loss of imprinting of these imprinted genes are associated with several human syndromes and with human cancer. Some of these human syndromes are, for example, Prader-Willi, Angelman syndrome, Silver-Russell syndrome, Beckwith syndrome, and all of them share different features like such as aberrant growth, mental retardation, cognitive problems, and one important thing is that they have common molecular findings that describe how these diseases are happening. Mainly epimutation or modification of the methylation state of these imprinting control regions are causing the diseases. Here I want to show you, this is the murine, the mouse genome, and chromosomes are all indicated, 19 chromosomes, not the sexual chromosomes. And you can see that in red, I'm representing the maternally expressed genes, the genes that are expressed only by the maternal chromosome. And in blue, the genes that are expressed, the imprinted genes that are expressed only by the paternal chromosomes. As you can see, they are organized in clusters. In these boxes represent one of the different imprinted clusters. As you can see, they are together in different regions of the genome, and they are mixed, maternally expressed and paternally expressed within the same cluster. Why are they organized in cluster and how? Here I try to represent a maternal and a paternal chromosome and the imprinting control region with a unmethylated in white region and a methylated region. As you can see, this is a DMR or a differentially methylated region, and this is what is controlling that maternally expressed genes are expressed only by the maternal chromosome and silent in the paternal chromosome, and on the other side is also controlling that paternally expressed genes are expressed only by the paternal chromosome and not by the maternal chromosome. What happens when we alter the methylation pattern of this imprinted control region? As you can see, that was the normal situation. If we remove methylation in the paternal chromosome, what it happens is that the paternal chromes becomes like a maternal chromosome. Then double dosage of the maternally expressed genes is occurring. The same, the opposite happens when I show the normal situation, and then if we add, or if the paternal chromosomes acquire methylation in the unmethylated region, what happens is that the maternal inherited chromosome becomes or looks like a paternal chromosome, losing maternally expressed genes. This imprinted control region is regulating the whole expression of all the genes within the cluster. Is why these genes are located in or are grouped in clusters. Now I'm going to jump a little bit to explain what is the cell model that we are working on, and then I will later explain all the experiments I want to show you today. We are working with the neural stem cell population in the neurogenic niches, trying to understand how this epigenetic process, the genomic imprinting, is regulating the behavior of these cells, and how aberration of imprinting participates in malignant transformation. So there are two neurogenic niches in the adult mammalian brain: the supranuclear zone in the hippocampus that is generating neurons, showed here in green with a GFP reported of the double cortin marker. So this in green, you can see all the neurons that are being incorporated in the hippocampus. And in the subventricular zone is the other neurogenic niche, and new neurons are being incorporated from the subventricular zone and incorporated to the olfactory bulb, where they integrate in the neuronal circuitry. Neural stem cells are in both niches. GFAP positive cells are astrocyte-like cells that self-renew and give rise to progenitors or type C cells that finally generate neuroblasts, oligodendrocytes, and astrocytes. The purpose of this line in our lab is understand how neural stem cells become malignant or acquire malignant transformation, becoming an aberrant neural stem cells that is known and described that can migrate and convert in cancer stem cells to form or to generate a tumor within the brain. This can migrate very far from the subventricular zone and generate different types of tumors. One of them is the glioblastoma, which is the most aggressive type of tumor in the brain. Here you can see, using a TDT tomato reporter, you can see different tumors that have been formed in different areas of the brain. So the hypothesis, the starting hypothesis of this work is to first to understand how genomic imprinting participates in this malignant transformation from the neural stem cell to the cancer stem cell. It's known that different mutations need to happen in these cells to become malignant. However, epigenetics, and more concretely genomic imprinting and loss of genomic imprinting, is one of the mechanisms that have been described in human cancer. So we are working on defining this process. The model that we are using to generate brain tumor from a neural stem cell, we are using a reprogrammable mice that contains a cassette, doxycycline inducible cassette expressing OC4, SOX2, KLA4, and CEMIC, the 4 Yamanaka factors. So giving doxycycline in the drinking water to the adult mice during a month, we can find we generate the induction of tumor formation. I'm going to show here how we are targeting neural stem cells, and more concretely the quiescent stem cell population in the subventricular zone to generate tumors. What we have done is to electroporate mice from these reprogrammed mice at postnatal day 2. We electroporate the mice with 3 plasmids. One is an episomic nesting CRE-RT2 plasmid that express chloride combinase inducible by tamoxifen, an integrative plasmid that contains GFAP reporter, but with a stop codon flanked by LOXP sites, and transposase to integrate this plasmid. We are then cells that retain the nesting CRE-RT2 plasmid after birth and during the 2 months that the mice become adult. only the non-proliferative neural stem cells, because it's an episomic plasmid, will be able, after tamoxifen injection, to induce GFAP or to promote GFAP expression, and then determine whether these tumors that we generate after doxycycline treatment are originated from neural stem cells. First, I want to show you an image of how we find this, the brains of these mice after doxycycline induction. We find regions in the brain that are vascularized and a bit altered anatomically. And when we extract these areas from the brain and check the expression of the cassette, the OC4, SOX2, KLI4, and CEMIC cassette, we find that this is overexpressed in tissue extracted from the brain and not from other regions from the brain, out of the brain, like liver. Sorry, I forgot to say that the doxycycline transactivator is under the promoter of the GFAP gene, so only cells that express GFAP are being induced by doxycycline treatment. Is why we never find expression in out of the brain, like you can see here in liver tissue. After the experiment, we find, well, we use control animals that do not contain the cassette, to see whether the neural stem cell, the quiescent stem cells, are being electroporated. And you can see that neural stem cells are located near the ventricle. But in the reprogrammable mice, we find these tumoral masses. You can see that a huge quantity of cells have been generated near the ventricle, that these cells are all GFAP positive, which indicates that come from the electroporated neural stem cells. And we find, you can see here by hematoxin eosin staining, that these tumoral regions are disorganized, and even when we cut the tissue, they break because they are very compact. So we are also able to extract, to isolate these cells, and to inject these cells in the flank of nude mice. And these cells are able to generate teratomas when these are injected in the nude mice, immunocompromised mice, suggesting that these cells have this tumorigeneity capacity after inducing the malignant transformation of the neural stem cells. When we, sorry, here I wanted to show you one of these tumors that have been formed after induction with doxycycline, and you can see that they express vimentin and also GFAP, indicating that they could be also, they can be like glioblastoma-like tumors. As I mentioned before, we isolated these tumor cells, also neural stem cells that were not induced to malignant transformation, and determined the expression of 40 imprinted genes by qPCR, and observed that 85% of these imprinted genes analyzed were altered in their level of expression. Here in this graph, you can see in green the upregulated genes, in yellow the downregulated genes, and down you can see that the paternally expressed genes in blue, the maternally expressed genes in red, and they are also organized by chromosomes, chromosome 1, 2, 6, 7, 9, and so on. So you can see a big number of imprinted genes are altered in their level of expression. As I said, 80% of the imprinted genes analyzed were modifying the expression. Now I want to focus on the chromosome 12. There is an imprinting cluster known as DLK1 and MEC3 genes, because it was one of the previously described imprinting cluster that can be involved in malignant transformation, because has been described that have a role in other type of tumors. So here is DLK1 is reducing the level of expressions in malignant in tumoral cells, and MEC3, that is the behavior gene, is upregulating the level of expression. Again, this is the representation of the imprinting cluster. Here is the imprinting control region with methylation, the differentially methylated region. In this case, the paternal chromosome is methylated and the maternal chromosome is unmethylated. How to understand whether these changes in expression is due to transcriptional regulation or if this alteration is due to the imprinting state of this imprinting cluster. Now I'm going to make a small break to explain how we in the field analyze the genomic imprinting state of cells or different tissues. It's not a simple thing to do, because we need to analyze the allelic expression of in different genes. So the maternal expression of the gene and the paternal expression of the gene. So to do this, we need to use hybrids using two different mice lines. For example, breeding black cis mice with castaneous mice, in which we have identified a single nucleotide polymorphism in the gene of interest. That means that in this gene of interest, we will find a nucleotide, for example, T in black cis and C in castaneous. Then, when we generate the hybrid offspring, the F1 hybrid offspring, if we analyze the neural stem cell population and the tumoral cells, what we will find, this is an example, what we will find is if we analyze DNA, so genomic DNA, we need to find the two peaks of expression, the two peaks of the two nucleotides for the single nucleotide polymorphism. This validates that using hybrids, we can analyze differential allelic expression of the gene. This is in genomic DNA, but what happens when we analyze the cDNA? The cDNA. We will need to generate what we call reciprocal crosses. One cross will be black cis mothers with castaneous fathers, and the other, the reciprocal cross, will be castaneous mother with black cis fathers. If we analyze the F1 of these reciprocal crosses and then analyze the cDNA, the mRNA, the expression levels, we will, for example, in this case, is a paternal expression Y, black cis is the mother, castaneous is the father. The father will have in the SNP a C, and when we sequence the mRNA, we find a C in this F1 offspring. If we analyze the reciprocal cross, the black cis is the father, and then it's a T. And when we analyze by sequencing the expression of this, sequencing of the gene, we find in this position that is a T. This will tell us that this gene, this example, shows paternal expression of the gene. What happens when we find a loss of imprinting? How we will identify this loss of imprinting? We will analyze mRNA or cDNA, and then we will find the expression of the two alleles, the two peaks of the C and the T. We can also use, more recently, we have indeed using the pyrosequencing technique to determine the percentage of allele expression for the different genes. Once we understand how we can identify the different expression of the two parental chromosomes, I'm going back to what I was saying, is that we are going to focus on the chromosome 12 and determine whether this change in expression is are due to transcriptional regulation of an alteration of the genomic imprinting within the cluster. Then we first analyze by sequencing the MEC3 gene, that is the second gene closer to the DMR. And using these reciprocal crosses that I told before, black cis castaneous and castaneous black cis. In this case, MEC3 is a maternally expressed gene, as you can see here. And then if it's a maternally expressed gene, if I analyze the SNP, I will need to find the T that is expressed by the black cis strain. And you find here in the neural stem cell population that is a T. In this case, when we analyze the castaneous black cis reciprocal cross, we will need to find if this is a maternally expressed gene. The C is the nucleotide that appears in the sequencing. This is confirming that neural isolated from these mice an expression of the gene, and everything is correct as we expected. Tumoral cells show a similar pattern of expression. Thus, they show a T and a C, suggesting that a maintenance of genomic imprinting for this gene is occurring in tumoral cells. Therefore, this increase of expression of MEC3 has to be caused by a transcription. What happens with DLK1, which is the other gene within the cluster near the imprinting control region? You can see here that, again, the reciprocal crosses, sequencing, and surprisingly, neural stem cells were showing a loss of imprinting of for this gene. We found C and T, the two allele expression of the gene, and in neural stem cells. Okay, so biallelic expression of DLK1 was originally observed in neural stem cells. When we analyzed and studied the tumoral cells, what we found was that if we pay attention to the sequencing for the tumoral cells, in this case, we found a T, which is correlating with the expression of the maternal allele. You see that is a T in black cis castaneous reciprocal crosses and a C in castaneous black cis reciprocal cross. That indicating that in this case, DLK1 shows in tumoral cells shows a maternal expression of the gene. If we go back to the imprinting cluster, DLK1 is a gene that is supposed to be expressed only by the paternal allele, and somehow tumoral cells alter the expression of the gene, the imprinting status of the genes, and become a maternally expression of the gene, showing that the imprinting state of DLK1 is altered in tumoral cells, showing a plastic epigenetic regulation in neural stem cells and during tumoral transformation. We next analyzed whether the methylation of the imprinting control region had something to do with the this modification of imprinting in NSCs and in tumoral cells. We performed this by pyrosequencing. That is a technique that allows you also to determine the methylation status of different CPGs within a specific region. Here you can find the imprinting control region that is also known as IGTMR for the DLK1 MEC3 imprinting cluster. You can see the CG, the CPG sites, and then we can sequence pyrosequencing and then analyze the level of methylation for these different CPGs. Here I have represented 10 CPGs within this region, and you can see that with the expected methylation in this case we have used brain tissue, will be 50%. Why? Because we find in the cluster that the paternal chromosome have 100% methylation and the maternal chromosome has 0% methylation. Thus, then the analysis will give you the mean methylation for the whole cluster will be then 50%. As you can see in black, brain tissue shows the expected 50% methylation in all of the CPG analyzed. When we study the neural stem cell population, you can see that the levels of methylation are increased, reaching 70%, 80% methylation in neural stem cells. This explains the acquisition of expression of the DLK1 expression in neural stem cells. So the biallelic expression of the gene in neural stem cells is due probably to the acquisition of methylation of the maternal allele. If we, as I mentioned before, if this IGTMR, the DMR acquires methylation, the maternal chromosome becomes a paternal chromosome, and then more levels of methylation of DLK1 increasing, also expression of the maternal allele for DLK1 will appear. That is what we find in neural stem cells. When we analyze again tumoral cells, the levels of methylation go down, decrease, and in this case, DLK1 becomes maternally expressed. We still don't understand how is this happening, but they lose methylation, and this makes losing the or acquiring the imprinting status again, but we still don't know how is this happening. Now, briefly, I would like to just in 2 slides to show you a new model that we have using to generate glioblastoma cells in mice brains. We use neural stem cells isolated from the P16, P19 deficient mice. We culture these cells and then overexpress the human EGF receptor with a GFP promoter reporter. And these cells are injected intracranially in the striatum of nude mice. And then they generate tumor glioblastoma tumors that can be isolated and grown in established, we can establish cell lines of glioblastoma cells. We have very recently done our RNA-seq with comparing the glioblastoma cells with the neural stem cells. And we have found, this is very recent, we find a huge alteration of the genome of the in glioblastoma cells. You can see that 11,000 genes are altered in glioblastoma cells. And when we focus on imprinted genes, we find that half of the imprinted genes are altered also in glioblastoma cells. This we are using also this model in combination with the reprogrammable mice to analyze how imprinting is being regulated. But we still don't know which genes are being modified in this model. So I'm going to conclude briefly that in vivo cellular reprogramming by this, the expression of the Yamanaka factors in brain cells leads to the formation of brain tumors. We are still defining whether these are glioblastoma tumors or other type of tumors like teratoma that will be the expected ones from the this model. We know we have seen that brain tumors formation involve changing several imprinted genes, and that the alteration of the imprinting state of DLK1 in tumoral cells suggests a plastic regulation of this genomic imprinting. And finally, only thank to all the members of the lab, PhD technicians and collaborators, Manuel Serrano, who is giving us the reprogrammable mice, Cristina Gil-Sanz, which is doing with us the electroporation, and Pilar Sanchez, who is doing the glioblastoma analysis, and all the funders. And that's it. That's all. I'm happy to take any question. Thank you.
Thank you.
Thank you.
Yes.
Yes.
Thank you very much. I have well, it's time to the discussion. You can raise your hand or sending a message by chat.
Eva, sorry, excuse me. Could you try without the earphones? Because the other day we had the same problem, and I think it was because of the earphone. I'm not sure, but
now it's correct.
Yes,
Yes.
it is. Thank you.
Okay. Then, any question?
Alguien ha levantado la mano, creo, Guillermo.
Where?
Hello.
Yes.
Hello,
Guillermo.
Guillermo from Complutense University.
Okay,
So-
go ahead.
So congratulations for very nice work and very nice talk. So I was just wondering, obviously, can you hear me well?
Yes, perfectly.
Okay.
Yes.
So I was wondering, obviously, genomic imprinting is very interesting, and I think you showed very nicely that there could be a contribution from the origin from this neural stem cells transformation. And I was wondering what could happen when the tumor progresses, because there are many genetic alterations and obviously also chromosomal rearrangements. So I don't know if there are any studies on how the genes that are imprinted are actually part of the mechanism by which the tumor continues growing, or because all the chromosomal alterations, this is even selected to have the chromosomes that are expressing these genes, so some information on the progress of the glioblastoma.
That's a good point, because so imprinted genes are mainly involved in fetal growth. So it's a epigenetic mechanism acquiring during placentation. So that means that they regulate, and it's like they contribute, the maternal and paternal information or genomic information contributes on how the embryo has to grow. That means, for example, DLK1 and MEC3 are compensating the growth of the embryo. It has been described that these are all growth regulators. So, for example, IGF2, I don't know if you know that IGF2 is an imprinted gene, and is known that is participating in how the tumor progresses and grows. So this is the hypothesis that they contribute to the growth of the tumoral mass. So I'm losing this control because, as I told you, because in the placenta is very important that imprinting is very well maintained. So alteration in placenta is that results in a big embryo, a big fetus. If this is regulating growth during development. So we believe that these imprinted genes are making the same thing, but controlling the growth. Alteration in the imprinting control alters the growth of the tumors. And IGF2 that is an imprinted gene, is already described that is regulating this.
Thank you.
Yeah.
Or questions?
So maybe I can ask another thing if you don't mind.
Thank you.
I think it was very interesting. So I was wondering, I mean, obviously you use these Manuel Serrano's mice that, as you discussed, is not really the perfect model for glioblastoma. But there are also other models. I mean, some of them are very complicated, but some of them are more or less affordable to try to develop gliomas out from the neural stem cell population. So are you thinking on maybe using some of these animals? Or I was thinking, for instance, in the nesting TDA model, where you can maybe induce the expression of some genes and maybe follow more or less easily.
Yeah, this is one of the more question points in the project. We are using this. So we use the Manuel Serrano's model because we have another line that is very focused on how acquisition of pluripotency is being regulated by genomic imprinting, and we found unexpectedly using these mice that they express CD133, and so it's not the expected model to use, I understand. And yeah, we are now trying to get other glioblastoma, more accepted models. But this is very interesting because understanding whether this malignant transformation has an intermediate process of like acquisition of some pluripotency was also important or interesting to us to address. But yeah, I know this is always a question that this is not the best model. I agree.
Very interesting, but maybe not. I mean, not the canonical for glioblastoma, but still very interesting. So
yeah.
Thank you.
Thank you.
No more questions? Then thank you so much, Zachary, for your very interesting presentation and all the audience. And see you in the next section.
Thank you very much for the invitation. Thank you.
Okay.
Bye.
Bye.
Bye.